Hi everyone, welcome to our chemistry tutor. Today we're going to be talking about pentose synthesis. I'll advise you after this video, you can go down to the channel and type pyrimidine synthesis alchemist tutor, which is the continuation of this video. Today we're going to be talking about the synthesis of pentose phosphate, how it is synthesized. We all know that our pentose sugar is known to be about five carbon sugar. So we have to synthesize it, and this is synthesized with the help of what? It is synthesized from what? 6 carbon sugar. We all know that glucose is known to be a 6 carbon sugar. So let's go with how it's with its pathway here. Here we have a glucose, which is the structure here. This is an alpha glucose. In various tissue in our system, we have different kind of enzymes and different kind of enzyme with different km value. The km value of each enzyme determine how that enzyme is going to catalyze our substrate. Having our substrate to be the glucose, and here our enzyme we are using to catalyze this reaction is known to be hexokinase. Hexokinase is known to have what? Higher km compared to that of glucokinase. Glucokinase and hexokinase are isozyme because they are what? They are enzymes that perform the same function. But the only difference in between them is that one has what? Higher km, and while the other has what? Lower km compared to the other. So this enhances what? The reactivity of that enzyme. So we prefer hexokinase in some certain tissue to another. So now we are having our hexokinase or glucokinase to catalyze this reaction of glucose by converting our glucose into what? Glucose 6-phosphate. How do we go with this procedure? This process requires energy. So how do we get our energy? We all know that our energy currency ATP, which is what? Adenosine triphosphate, is our energy currency which we use in this process to overcome this common reaction to have our byproduct. And having our ATP here, a molecule of the phosphate in the ATP, adenosine triphosphate, a molecule of the phosphate is used it is used in this reaction, and it is joined with what? With our glucose, having our end product here to have what? Our ADP, which is adenosine diphosphate. Now the phosphate group that we have on this ATP, a single phosphate group has left this ATP, and this is a way of cleaving our ATP, cleaving the phosphate from the adenosine triphosphate. The process that we cleave that phosphate from the other phosphate conjoined to what? Our nucleoside. It enhance and bring out what? Higher energy to catalyze this reaction. That is why we have our what? Our phosphate conjoined with what? With the glucose. That's why we're having what? Our glucose, which is what? Attached to our phosphate attached to the glucose on carbon 6, which is situated in this place. Now having our glucose 6-phosphate, if we could remember the pathway of glycolysis, we know after this process we do have an enzyme that catalyzes this process, this step, to give us another step of fructose 6-phosphate, which is known as what? Isomer. But that is not where we are going today. Today we have to what? To apply an enzyme, since we already know we are going to produce our pentose, which is known as what? A 5-carbon sugar, which is what? Pentose. Now we have to know a way of cleaving out a carbon from this hexokinase, which is glucose, which is known to be what? 6-carbon sugar. We have to have different enzymes in our body system that enhance this procedure. Now we have glucose 6-phosphate dehydrogenase. What is the function of this dehydrogenase? The function of this dehydrogenase is just the removal of what? Of an hydrogen from glucose 6 phosphate. And how do we do that? We need to introduce our co-enzymes, which is what? NADP plus, and that NADP plus is input into that reaction, and it takes out and bring out the hydrogen from this glucose 6 phosphate, and we have here our NADPH. And what do we have left? Our byproduct is now 6-phosphoglucanolate, and this is what? A ketone. The product we have here is what? Is a ketone. Nevertheless, what we need to get here is not a ketone. We need to get what? A glucose. But during this process, we have to what? To get into different places or different steps which are not the what? The main intermediates in this reaction. So we continue the reaction in our body system. We have to produce what? Another enzyme which is known to be what? Lactonase. What does this enzyme do? ACE, that is the lactone. We have to remove what? The lactone from what? From this product. That is the function of this lactonase. I have explained previously that whenever you see any compound that end with ACE, that is, it is acing the reaction. That is, it is even telling us this is what? Is an enzyme. Now we have this enzyme lactonase that has as what? As a precursor that catalyze this reaction and remove the lactate from this phosphoglucanolactone. And what is our byproduct? And in this process, we need to input in what? We need to input in water molecule. And after we input in water molecule, what do we need in that? We need the OH group in that water molecule. And hydrogen is passed out from this reaction. Now, what do we get now? We are going to end up getting our 6-phosphoglucanolate. In the next step now, after producing this in our body system, another enzyme comes in, which is what? 6-phosphoglucanate gluconates dehydrogenase, just like in the procedure we have in step 2. In this step 2, we need a dehydrogenase for us to remove an hydrogen from this compound. The same thing is applicable here. We need what? A dehydrogenase of what? A dehydrogenase of phosphogluconate, and this coenzyme or cofactor is introduced to take out the hydrogen. Here we have our NADPH after this reaction. And what do we have next? We have a ribulose 5-phosphate. This ribulose 5-phosphate is a 5-carbon sugar, but it is what? Is a ketone. Is part of what? The ketones. When you're talking about the ketos, we have the ketos and what? We also have the aldose. I'm talking about the aldose. Aldose are what? Are sugar made from what? From aldehyde. Example are the glucose. Example are the ribose, etc. And we also have the what? Ketos. The ketos are sugar made from what? From ketone functional group, which are all gotten from a ketone functional group. Example are fructose. Example are ribulose, etc. like that. Now, this is not what we what we intend to synthesize initially. Our main target is what? Is pentose phosphate synthesis. Is what? Pentose phosphate synthesis. But here we only have a pentose, although this is a pentose, but this is a ketose. So we have to change this ketone into what? Into an aldehyde. How do we do that? We need to introduce another enzyme which is known to be what? The isomerase of this structure. That is, what does this imply? It means that just restructuring of our compound ribulose 5-phosphate. You just have to restructure the what? The functional group. The functional group. And we know that the functional group we have to have in our ribulose 5-phosphate is what? Is a ketone. And what we intend to produce is an aldehyde functional group. So we introduce an enzyme which is known to be what? Isomerase, to catalyze this reaction, and we are going to get ribose 5-phosphate. Yeah, we've gotten to this point now, but before this could be used in uh in nucleotide synthesis, it has to be in what, in a cyclic form, and we need to introduce another enzyme, which is what, ribophosphate pyrophosphate kinase, and the enzyme is denoted with RPPK. RPPK, which is what, ribophosphate pyrophosphate kinase. That is the function of the enzyme. So what do we get after this? We are going to get PRPP. We have to get phosphoribosylpyrophosphate. Phosphoribosylpyrophosphate. This is now the end product of our synthesis of pentose phosphate, of our pentose phosphate, which can be used in what, it can be used in our nucleotide or nucleotide synthesis. Please, all right, if you are watching this video today, please enable to like, subscribe and comment. If you're having any issue and any cause or what we've just explained that is yet to get clear to you, just type in the comments and you shall see a solution. Thank you.